Hi everyone, Dr. Rashid here. So today I will be explaining you the metabolism of some minerals in our body. So first of all, you should know the difference between micro minerals and macro minerals. So on the basis of nutritional requirement, the minerals are classified into macro minerals and micro minerals. For macro minerals, their daily requirement is more than 100 milligram. So examples for macro minerals are calcium, phosphorus, sodium and potassium. And in case of micro minerals, their daily requirement is less than 100 milligram, and the examples are iodine, fluoride, iron, copper, zinc and selenium. So this classification is on the basis of nutritional requirement. So first of all, we will see the metabolism of calcium. And regarding calcium, the total amount of calcium present in our body is around 1 to 1.5 kg. So among this 1.5 kg, 99% is present inside the bone and teeth, and the remaining 1% is present in the body fluid, including the blood. And the normal blood level of calcium is around 9 to 11 milligram per deciliter. Coming to the dietary sources of calcium. So milk is the richest sources of calcium, and the other sources are egg, fish and vegetables. And regarding the nutritional requirement, an adult individual requires almost 500 milligram of calcium per day. And in case of children, the requirement is around 1200 milligram per day. And in pregnancy and in lactation, the requirement is more, so almost around 1500 milligram per day. And coming to the metabolic functions of calcium, the major function is calcium is required for the calcification of teeth and bones, that is mineralization process. So this mineralization maintains the tensile strength of teeth and bones. And the second function is they are related to coagulation, that is this function is related to coagulation. Calcium itself considered as a clotting factor 4. So calcium have an important role in thrombin formation. And the third function is muscle contraction. So during muscle contraction, this calcium is released from sarcoplasmic reticulum, and this calcium binds with one of the muscle protein, that is troponin C, and that process initiates muscle contraction. The next function is calcium also have an important role in nerve conduction. So calcium is necessary for the transmission of nerve impulse. So the presynaptic nerve contains presynaptic vesicle, and calcium is essential for the release of neurotransmitters from this presynaptic vesicle. So calcium is essential for the nerve conduction. And calcium is also essential for the functioning of hormone. Some hormones, especially those hormones binding with cell membrane receptor, they require second messengers. And calcium is also considered as one of the second messengers in signal transduction. And regarding vascular permeability, calcium decreases the passage of serum through the capillaries. And calcium is also required for the activation of certain enzymes, especially the kinase group of enzymes. So these are the enzymes which require calcium for their activity: adenyl cyclase, calcium dependent protein kinase, calcium magnesium ATPase, glyceraldehyde 3-phosphate dehydrogenase, glycogen synthase and myosin kinase. So these are the calcium dependent enzymes. And concerned with the myocardium, calcium prolong the myocardial contraction, that is they prolong the systoles. So these are the different functions of different biochemical role of calcium. Coming to the normal blood level of calcium, the normal blood level of calcium in an healthy individual is around 9 to 11 milligram per deciliter. And let's see how this normal calcium blood level is regulated. So these are the factors affecting calcium homeostasis. One is vitamin D, the second one is parathyroid hormone, and the third one is calcitonin. So these are the three components which maintain the normal blood level of calcium. Here vitamin D and parathyroid hormone increases the blood calcium level, whereas calcitonin decreases blood calcium level. Coming to the functions of vitamin D, vitamin D increases the absorption of calcium through gastrointestinal tract, and in kidney this calcitriol increases the reabsorption of calcium through the renal tubule, and in bone this calcitriol increases bone mineralization process. So the net effect of this vitamin D is they increases blood calcium level. And regarding parathyroid hormone, in kidney this parathyroid hormone increases the reabsorption of calcium, whereas they decreases the reabsorption of phosphorus, and in gastrointestinal tract they increases the absorption of calcium. So the net effect of this parathyroid hormone is they increases blood calcium level. And in bone this parathyroid hormone increases bone resorption process. And coming to the function of calcitonin, in bone they inhibit bone resorption, and in kidney they decreases the reabsorption of calcium as well as phosphorus. So the serum calcium level is maintained by calcitriol, parathyroid hormone and calcitonin. Calcitriol and parathyroid hormone, they both of them increases plasma calcium level, whereas calcitonin decreases plasma calcium level. So that's all regarding calcium. The second important micromineral is iron. Coming to the total body iron content, our body contains 3 to 5 gram of iron. Among this, 75% is present in blood, and remaining 25% is present in liver, bone marrow and in muscles. And just remember this protein name, that is one is ferritin, that is the storage protein for iron. So ferritin is the storage protein for iron, and transferrin, that is the transport protein for iron. Regarding daily requirement, an adult individual require 20 milligram of iron per day, and children require 20 to 30 milligram of iron per day, and during pregnancy and lactation the daily requirement is 40 milligram. Coming to the sources of iron, the richest sources are green leafy vegetables and jaggery, and the other sources are pulses, cereals and meat, and milk is a poor source of iron. And there is another important exam question, that is iron absorption and the factors regulating the iron absorption. And iron absorption occurs through the proximal part of the duodenum. So the site of absorption is duodenum. And iron having two form, that is reduced form, and reduced form is also known as Fe2 plus form or the ferrous form, and the oxidized form of iron is Fe3 plus or ferric form. And our food items contain the iron in the form of oxidized form, that is the ferric form, but absorption occurs only through ferrous form. So ferric iron is present in the food items, and this one is the intestinal epithelial cells, and this intestinal epithelial cell surface contain some reductase enzyme. So this reductase enzyme reduces this Fe3 plus iron into Fe2 plus iron, and there is a protein channel which is present in the epithelial cell surface, that is the apical surface of the intestinal epithelial cell, that is known as divalent metal transporter 1, that is DMT1, because they transport divalent cation. So Fe2 plus is a divalent cation. So through this divalent metal transporter, these ferrous ions are transported across the apical surface of the duodenal cell, And once they reach the interior of the duodenal epithelial cells, this ferrous ion have two metabolic fate. If our body has sufficient iron, that absorbed ferrous ion is stored within the intestinal cell as ferritin. That is the one of the fate of this absorbed iron, right? And the second metabolic fate is they are absorbed through the basal surface of this epithelial cell and to reach the blood. And this is mediated by another protein which is known as ferroportin. So there are two protein channels which is required for the transport of this iron. One is divalent metal transporter 1, which is present in the apical surface of the epithelial cells, and the second protein channel is ferroportin, that is present in the basal surface. And this transport occurs in the form of ferrous ion. So once this ferrous ion reaches the blood, this blood contains some oxidizing enzyme, and the best oxidizing enzyme is ceruloplasmin, and this ceruloplasmin having ferroxidase activity. So ceruloplasmin convert this ferrous ion into ferric ion, and this ferric ion is transported through this transferring protein. So ferric ion is present in the food item, that is the oxidized form of iron, and this ion is reduced into ferrous state and it is attached to the divalent metal transporter on the mucosal surface, and this ferrous ion is internalized. This ferrous ion within the epithelial cell having two metabolic fates. One is the ion is oxidized into ferric form, which binds with apoferritin and it is converted into ferritin where it is stored. The second fate is they crosses the cell membrane through another protein, that is ferroportin protein channel, and they finally reaches the blood where it is oxidized into ferric state by ceruloplasmin, and this ferric ion is bound with transferring protein. And regarding factors increasing absorption, one is acidic pH. Acidic pH promote iron absorption, and the second component is vitamin C or ascorbic acid, because vitamin C having strong reducing property. So due to their reducing property, vitamin C help in the conversion of ferric ion into ferrous ion. Regarding factors which decreases iron absorption, these are oxalate or phytate. If oxalate or phytate components are there in the food item, they form an insoluble complex with iron. Thereby, they can interfere the iron absorption. Regarding regulation of iron absorption, there is a theory, that is mucosal block theory. According to this theory, the iron absorption, the regulation occurs at the level of absorption. That's why it is known as mucosal block theory. So there are two protein channels which is required for the iron absorption. The first one is divalent metal transporter and the second one is ferroportin. And these two proteins are regulated by another protein that is known as hepcidin, which is synthesized by the liver cell. And this hepcidin function is they promote the degradation of DMT1 and ferroportin. So whenever there is a sufficient iron in our body, our liver cell produces this hepcidin protein, and this hepcidin protein promote the degradation of divalent metal transporter 1 and ferroportin. Thereby hepcidin protein inhibit iron absorption. That theory is known as mucosal block theory. And regarding transport of iron in blood, this is mediated by a protein that is known as transferrin protein. So transferrin is the transport protein of iron in blood, and one molecule of transferrin carry 2 ferric ion. And regarding ferritin, that is the storage form of iron, and one molecule of ferritin carry around 4000 ferric atoms. And regarding iron deficiency, these are the different causes for iron deficiency. The first one is the nutritional deficiency. So dietary deficiency of iron rich food items produces iron deficiency. The second one is hookworm infestation, repeated pregnancy, and if there is a defective absorption of iron, that can also produce iron deficiency, and chronic blood loss. So all this produces iron deficiency. Coming to the clinical manifestation of iron deficiency, the patient is usually the most common clinical presentation is fatigability. And regarding clinical signs, the patient will be having tachycardia and palpitation. In severe iron deficiency, there will be brittle and spoon-shaped nails. And peripheral blood smear shows microcytic and hypochromic anemia. That is, peripheral smear shows microcytic and hypochromic anemia. That will be the peripheral smear picture in iron deficiency anemia, microcytic and hypochromic. Regarding treatment, that is the oral iron supplementation. So that's all regarding iron. And the next important mineral is copper. And in copper, you should know what are the copper containing enzymes, what are the biochemical functions of copper, and metabolic disorders related to the copper. Coming to copper containing enzyme. These are the copper containing enzymes. One is ceruloplasmin, the second one is ALA synthase, lysyl oxidase, tyrosinase, and superoxide dismutase. These are the copper containing enzyme. So let's see the functions of this enzyme, and that's constitute the biochemical functions of copper. Regarding ceruloplasmin, ceruloplasmin is a protein present in our blood, and this ceruloplasmin having ferroxidase activity. So this ceruloplasmin is a copper containing enzyme. So copper is essential for iron absorption. So copper deficiency can produce iron deficiency. Coming to ALA synthase. ALA synthase is a major enzyme in heme biosynthesis. So hemoglobin formation require ALA synthase enzyme. So copper deficiency can produce heme deficiency, thereby anemia. Coming to lysyl oxidase enzyme. So the collagen proteins are strengthened by some covalent cross-linking. These covalent cross-linkings are present between lysine, hydroxylysine residues. And these covalent cross-linkings are produced by a particular enzyme which is known as lysyl oxidase enzyme. And this lysyl oxidase is a copper containing enzyme. And regarding tyrosinase enzyme, is an enzyme involved in tyrosine metabolism. And this enzyme is concerned with melanin formation from tyrosine. So copper is essential for melanin formation. So copper deficiency can produce hypopigmentation. And the last enzyme is superoxide dismutase, and this enzyme having antioxidant property. That's why copper is having antioxidant property. So these are the different biochemical functions of copper. Coming to copper deficiency manifestation, copper deficiency produces anemia. This is due to defective ceruloplasmin function and defective heme formation. And the second feature is hypopigmentation. It is due to defective formation of melanin from tyrosine. And the third clinical feature is aneurysm and rupture of aorta. And it is due to defective collagen cross-linking formation due to defective functioning of lysyl oxidase enzyme. And defective collagen cross-linking can lead to recurrent bone fractures. So all these are the deficiency manifestations of copper. Coming to the metabolic disorder. So Wilson disease is an inborn error of copper metabolism. So it is a genetic disorder, and the basic defect is it is due to mutation in the gene coding for a protein which is known as copper binding ATPase protein. That protein is present in the liver cell, and that protein function is this protein is required for the excretion of copper from the liver cell. That is the function of this copper binding ATPase present in the liver cell. So in Wilson disease, there will be mutation of that gene coding for that particular protein. So there will be defective excretion of copper from the liver cell. So copper will be accumulated within the liver cell. That will lead to liver cell injury. So once this copper concentration exceeds this liver threshold, that copper will be secreted into the circulation, and through this circulation, through this systemic circulation, this copper reaches different tissues in our body. So copper will reaches kidney where it produces renal damage, and copper will reaches the central nervous system where they produces neuronal degeneration, and copper will reaches the cornea where they produces KF ring formation. So these are the different clinical features of Wilson disease. So deposition of copper is increased particularly in organ like liver, brain, kidney and cornea. So the accumulation of copper in liver produces liver cirrhosis, and in deposition in neuronal cell produces neuronal degeneration, and their accumulation in kidney produces renal failure, and copper deposition in cornea produces Kayser-Fleischer ring. This is Kayser-Fleischer ring, a brownish ring around the cornea of the eye. So these are the different clinical features of Wilson disease. So that's all regarding copper. Coming to the next mineral, that is zinc. So regarding biochemical functions of zinc, you should know some example for enzymes containing zinc as a cofactor. So these are the zinc dependent enzyme. One is ALA dehydratase, and that enzyme is required for heme biosynthesis, and the second one is alcohol dehydrogenase, lactate dehydrogenase, and alkaline phosphatase. So these are the zinc dependent enzymes, and this zinc is having role in insulin storage. So when this zinc binds with insulin, that will promote the stability of insulin molecule. So zinc promote the stability of insulin molecule. So commercially available insulin preparation contain zinc. And the antioxidant role of zinc. There is an enzyme superoxide dismutase, is a zinc containing enzyme. So zinc is having antioxidant property. And zinc is required for spermatogenesis. That's why zinc is said to have an important role in reproduction. And zinc is also required for wound healing process. And there is a zinc containing protein which is present in our saliva, that is known as gustin, and this gustin protein is required for the activity of taste bud. So zinc is required for taste sensation. And protein biosynthesis, an enzyme which is RNA polymerase enzyme, and this RNA polymerase enzyme contains zinc. So zinc is said to have an important role in protein biosynthesis. So that's all regarding the different biochemical functions of zinc. Coming to the next mineral, that is selenium. Coming to the metabolic functions of selenium, selenium is one of the component of an amino acid which is known as selenocysteine, and this selenocysteine is considered as the 21st amino acid. So selenium is required for protein biosynthesis. The second function, that is the major function of selenium, that is antioxidant function. There is an enzyme which is known as glutathione peroxidase. It is one of the important enzyme which is having antioxidant property, and that enzyme contains selenium as a cofactor. And this function of selenium is complementary to that of vitamin E, and vitamin E is a fat soluble vitamin having antioxidant property. So availability of vitamin E reduces this selenium requirement, both having the same function, that is antioxidant property. And selenium is also required for the conversion of T4, that is tetraiodothyronine, into T3, that is triiodothyronine. So T3 is the physiologically active form of thyroid hormone. So in peripheral tissue, this T4 is converted into T3. This is done with the help of an enzyme known as deiodinase, and this deiodinase enzyme requires selenium. So selenium deficiency can produce as hypothyroidism. So these are the different biochemical functions of selenium. Okay, thank you.